Hello students, this is Faizan Mirza. We are discussing the topic of meiosis. A lot of students approach me to elaborate onto this topic, especially the stages of meiosis which lead to independent assortment of chromosomes. Somehow, when you learn about meiosis, the most difficult thing to comprehend at time becomes the independent assortment of chromosomes. So let's quickly go back to some facts of meiosis and we'll start our discussion and we'll end up to the very fact that how independent assortment occurs. So what's meiosis? We'll be discussing meiosis and independent assortment in detail in this video. To understand the basic concept of meiosis, you have to align the concepts of cell cycle, chromatin, chromatid, chromosome, DNA replication, homologous pair, gene locus. So these concepts must be in place accordingly to understand what's actually happening in meiosis and how it's actually connected with independent assortment of chromosomes and the genes and the alleles. So let's start our discussion of cell cycle. Recall cell cycle in eukaryotes. In eukaryotes, we know that the cell cycle is having three stages. Their first stage is interphase, which is followed by the nuclear division, which can occur by mitosis or meiosis either. And then you have the third stage, cytokinesis. Interphase is the stage where the cell is growing. There are three subparts, G1, S and G2, gap 1, semi-conservative replication in the synthesis stage, which is S phase. This is the point where the DNA starts to undergo replication, and then G2, gap 2. All these three stages are part of interphase where the cell is just growing and metabolically very active. DNA exists as chromatin, which appears granular if you look at the cell in microscope or in a photomicrograph. So whenever a cell is in interphase, any of these stages, be it before replication, be it after replication, or any part within the interphase, DNA will always exist as chromatin granular. Nuclear division could either be mitosis or meiosis. In mitosis, clones are produced, whereas in meiosis, gametes are produced. In mitosis, two diploid cells form eventually, and in meiosis, most of the times, four haploid cells form. Whenever you are following cell division or nuclear division, most appropriately, so when you look at a cell undergoing nuclear division, the DNA exists as chromosomes, either as single chromatids or two sister chromatids attached at the centromere. So this concept must be in place that how the DNA looks like during interphase and during nuclear division. So DNA exists as chromatin, granular in interphase, and exists as chromosomes during nuclear division. The chromosomes, initially they look like single chromatids, and then eventually two sister chromatids attached at the centromere, and then again as the cell divides, so it again splits. What's cytokinesis? Cytokinesis is the equal division of the cytoplasm into two daughter cells. This is not just the division of the cytosol, which is a liquid part of cytoplasm, but the organelles as well. Now, at this point, if you look at the cell, the DNA starts to uncoil to become chromatin again, so that after cytokinesis, the cell can go back into interphase and then spend the life in these three stages, where the DNA will exist as chromatin, then come to nuclear division, where it becomes chromosomes, and again cytokinesis, where they uncoil and become chromatin again. So, to move on, let's discuss what's chromatin and what's chromosome, and how these two terms are connected. Chromatin. Whenever you have DNA alone in a double helix, it appears granular and you call it chromatin. Here, the DNA is not condensed, so you don't really see thick DNA or thick chromosomes in the characteristic appearance. Rather, they appear granular, and this is all along the interface of the cell cycle. During S phase, this DNA will separate its two strands. The original DNAs, I'm showing this in purple color, so they separate out, and the green two new strands are made against the template strands by semi-conservative replication, something which I am sure you already are aware of. So DNA replication occurs by semi-conservative mechanism where the two strands separate, and complementary base pairing occur which allow two new strands to be synthesized against the template strands. The DNA coils up in such a way that one strand is the original strand and the other is the newly synthesized strands, something that was explained by the Meiselsons and Stahl's experiment. The two DNA molecules with the same sequences of bases will be formed, and all this would occur already when the cell is in interphase, and yes, still all the cell DNA will still be in chromatin. So this thing entirely is occurring in interphase. What happens in early prophase? In early prophase, the DNA starts to condense. It starts to wind around histone proteins, and the DNA thickens, or we say that the DNA condenses. This is the feature of the DNA that we end up having. A thick filament-like structure, and the DNA, two strands are here with these yellow dots. I have tried to just show these yellow dots. These are the histone proteins. So, and this is the centromere right in the center. So this is the chromosome, which actually you can see in early prophase. So chromosomes appear as single chromatid. But soon, this is the characteristic appearance of a chromosome where you end up seeing a chromosome having not one chromatid but two sister chromatids. So the chromosome appear as two sister chromatids attached at the centromere. Both sister chromatids, they have the same copy of DNA molecule which was replicated in the interphase. Hence the same sequence of bases on this particular chromatid and the sister chromatid. So in early prophase, the sister chromatids will always have the same sequences of bases. Another rule to keep in mind is that the number of chromatids is equal to the number of DNA molecules. If you can count the number of chromatid, it means you technically are counting the number of DNA molecules. Because one chromatid means one DNA molecule. Here there are two chromatids, so this means that there are two DNA molecules here. So if we are discussing a human cell, so when the cell is in interphase, a human cell, initially it is having just 46 DNA molecules. After replication, the DNA molecules become 92. So these 92 DNA molecules in interphase, they are not in the form of chromatid or chromosome as yet. All 92 DNA molecules exist as chromatin only. Now come early prophase. Again, 92 DNA molecules, they are as 92 chromatids on 46 chromosomes. And this is when early prophase begins. The chromosomes are seen to be in homologous pairs because we are a product of sexual reproduction. What's a homologous pair? So let's align this concept now. Chromosomes occur in homologous pairs. Homologous means that the chromosomes, they will have the size which will be same in the pair, the order of the genes will be same, the number of genes will be same, and the gene locus will also be the same. When you band them with a certain stain, they will give you a same banding pattern. One partner of each pair, each homologous pair, comes from one parent who was involved in the sexual reproduction. And the other partner of the same homologous pair of chromosome comes from the other parent involved in the sexual reproduction. So, because of this, sexual reproduction we have chromosomes in pairs. 23 chromosomes having 23 DNA molecules. They are coming in one gamete, and 23 chromosomes having 23 DNA molecules, they are in the other gamete. When these two gametes fuse together, zygote forms, having 46 chromosomes with 46 DNA molecules. So these 23 DNA molecules were actually like this in chromatids. 23 of these, this size, this size, this size, this size, and this size. And similarly, this gamete had 23 of these, similar of these. So this, the same of this. So again, 23 of these. Eventually, in this zygote, these chromosomes will form pairs. When this cell starts to divide, when this cell grows, eventually this cell will start to divide as well, so that this zygote can form 2 cells and then 4 cells. It continues division by mitosis. So these are the pair of chromosomes, homologous pairs. One chromosome for the other, one chromosome for the other, and so on and so forth. So how many pairs do we have? We have 23 such chromosomes in our somatic cells, 23 such pairs rather, and 22 pairs actually are of autosomes, and the 23rd is XX or XY, the sex chromosome. Alleles of the same genes always occupy the same gene locus on homologous pairs. Whenever we say a homologous pair, it means that the chromosome, both the chromosomes are exactly alike. So in females, there are 23 homologous pairs. The 22 pair of autosomes, and the 23rd chromosome pair is also a pair because X chromosome is there together with another X chromosome. So in female, there are 23 homologous pairs. We call them as 22 pairs of autosomes and the 23rd XX pair. In males, however, there are 22 pair of autosomes very similar to the females, but the 23rd is not a pair. So the 23rd is the sex chromosome XY. We don't call this as a pair because X is a different chromosome, Y is a different. They are not a homologous pair. But this is the chromosomes. These are the chromosomes XY which are there in males. So males are 22 XY, females are 22 XX. To go further, we have to understand a concept of karyotype. What's a karyotype or a karyogram? You take a photograph of a cell undergoing mitosis and is in the stage of metaphase. The chromosomes are thick and swollen. These can be taken into a photograph, and the photograph is then cropped and the chromosomes are pasted on a separate sheet in pairs. So the largest chromosome comes first, then the smaller, then the even smaller, and then the smaller one, and you just keep on decreasing there. You just arrange them in the order of decreasing size. So chromosomes can be placed in pairs in a karyotype. So chromosome, this is the human chromosome 1, the pair 2, the pair 3, pair 4, pair 5, pair 6, pair 7, 8, 9, 10. For example, if I say chromosome 12 of human beings, so this means that this is the chromosome 12. Either I'm talking about this chromosome or this chromosome. One of them came from the father, the other of them came from the mother, and probably if this is a chromosome 16 pair, so this probably came from the mother, this probably came from the father. We can't just say by looking at it which came from which parent. So this is the karyotype. In karyotype, we arrange a chromosome in the order of decreasing size and the position of the centromere. So we see the homologous pairs, and we place the sex chromosome at the last. So you can see by looking at the karyotype that this karyotype belongs to a male because the 23rd here chromosomes are XY and not XX. So you can see that Y is such a tiny chromosome as compared to X. So the alleles which are present, the gene locus which are present on the X chromosome are actually absent on the Y chromosome. So Y chromosome is only having the genes, the traits required for the development of sexual characteristics in males, or the masculine features of development. Karyotype can also be found, can also be displayed in this way, where the pairs are not shown, rather the chromosomes are shown singly. The other partner of the pair is not shown. So in this way, you can see that all the 23 chromosomes have been lined up. These are the 22 autosomes, and this is the X and Y chromosome. So all these chromosome 1 till 22 are the autosomes, and this is the XY chromosome. We group them in group A, B, C, D, E, F, G, depending on the size of the limbs of the chromosome and the position of the centromere. When you're looking at a karyotype, what use can it give you? You can't look for a genetic mutation on a karyotype, but you can look for a chromosomal aberration. What do you mean by the chromosomal aberration or a chromosomal change or a mutation? For example, if you have a karyotype here and rather than getting 46 chromosome, you get 47 chromosome. That means that the organism or that individual is suffering from a disorder where one extra chromosome is present in his or her cells. So that individual is 46 plus 1, which is 47 chromosome. And similarly, you may find a karyotype of an individual with 45 chromosomes, which actually is lacking one chromosome. So yes, these conditions of monosomy and trisomy can be seen here. This diagram shows to you the placement of the genes, a few of these genes locus on the human X chromosomes. So you can see that this gene here is actually enabling the kidney to retain phosphate. This one controls the production of your membrane protein found in the muscles, and you can just go through here all these proteins here, these genes here. This one is required for normal color vision. This produces the pigment, and this thing here is the one which actually is involved in the red-green color blindness as well. And we study factor VIII. We study how factor VIII is the gene which can cause hemophilia in males. So, and then hemophilia B as well can be caused by yet another protein which is another factor which is a factor 9 here, which goes missing. So this is sex chromosome, this is X-linked. So any trait which is present on this X chromosome, males will not have an alternate of that. Males will always have just one copy of all these. They do not have the alternate on the Y chromosome. Whereas females have two X chromosomes, so they have two alleles of these traits. So they can be homozygous dominant or heterozygous dominant or homozygous recessive for all these traits. Whereas for a sex-linked trait, a male, the males will always be either dominant for the trait or recessive. We cannot use the term homozygous for the sex-linked trait in males. We can't use the term heterozygous dominant either. So let's move on to meiosis. Meiosis I separates the homologous pairs. And the homologous pairs are separated into two daughter cells. The cell already had 92 DNA molecules as already covered previously. Both the daughter cells, they would receive 46 DNA molecules each, which means that the DNA content gets halved. The DNA will appear as characteristic chromosome. So this is early prophase. You can see the cell membrane, you can see the centrosomes. They have already started moving to the opposite poles. You can see the nuclear envelope, and these are the chromatids. You can see that the DNA is appearing as chromatids. The chromosomes appear as single chromatids. I am showing you two purple and two blue here. The chromatin, it starts to condense into chromatids here, and the nuclear envelope can be seen, the nucleolus can be seen, you can see cell membrane, and all these labels can very well be seen. What happens in middle prophase? That's middle prophase 1. So what's happening in middle prophase 1? In middle prophase 1, you have the chromosomes getting into their characteristic appearance, and in this way you can see that the chromosomes are appearing in pairs. They can be seen to be having sister chromatids attached to the centromere now. So homologous chromosomes pair up. Each pair is called as a bivalent. You can still see the nucleolus. What happens after that? We move on to late prophase 1. Here, the nuclear envelope appears to disintegrate. It starts to disappear. The bivalents, they come so close that crossing over occurs and chiasma forms. The nucleolus also starts to disappear. You can see that the centrosomes have moved to the opposite poles and spindle fibers have started growing. By late prophase 1, this is very well visible. What happens in crossing over? Crossing over, new allele combinations form because of crossing over. And why is that so? For example, this is a homologous pair, a bivalent. It undergoes crossing over. So this is how its segments will break and rejoin in such a way that the chiasma form between non-sister chromatids of the bivalents. And the non-sister chromatids, once they have these regions overlap, they exchange their parts. And as a result of exchanging their parts, new allele combinations form. By the end of prophase 1, nuclear envelope and the nucleolus completely disappear. And spindle fiber is fully formed. The spindle, the microtubules have fully formed. What happens after that is metaphase 1. In metaphase 1, these chromosome pairs, these bivalents, I am showing a crossing over successfully in one pair and not the other. So this is the pair that crossed over successfully. This is the pair that did not cross over. So, and you can see these segments have exchanged these segments. So spindle attached to the kinetochore on the centromere of the chromosomes, and these are the bivalents. They align along the equator of the cell. In anaphase 1, these spindle, they retract and the bivalents separate, the homologous pairs separate, and that's actually what's happening in meiosis I. The homologous pairs, they separate out. And the chromosomes, when they are pulled towards the centrosome, they are pulled centromere first. So the centromere can be seen going, whereas the limbs of the chromosome seem to be dangling along. What follows is telophase 1. In telophase 1, we see that the nuclear envelope starts to reappear and eventually it's fully formed. The spindle is breaking here. These are the remnants of the spindle which was formed initially. The nuclear envelope fully is formed around the haploid nucleus now. Haploid, why? Because the homologous pairs have been separated. One set of chromosomes in one haploid nucleus, and you can see the crossed over segment which is present here in this new chromatin, which is the new combination here. So the new and you can see another haploid nucleus here, again having a crossed over segment in that chromatin. Cytokinesis follow, and eventually, by the end of meiosis I, two independent cells can be seen and both have haploid nucleus. In animals, two haploid cells are produced at the end of meiosis I. But in plants, this doesn't occur in the very same order. In plant cells, the cell would go to meiosis II directly after telophase. So this stage cannot be seen. The cells separated, this stage doesn't occur. So meiosis, right after telophase 1, meiosis II proceeds. So the cell enters into prophase 2 right away. Both cells here have received 46 DNA molecules on 46 chromatids and 23 chromosomes. So the number of chromosomes have halved, the number of chromatids have halved, the number of DNA molecules have halved by the end of meiosis I. Now begins meiosis II. In meiosis II, the facts that you need to keep in mind is that the two daughter cells of meiosis I, they enter meiosis II. Each cell is having 46 DNA molecules on 46 chromatids appearing as 23 chromosomes. What's happening in prophase 2? This is exactly like mitosis now. Prophase 2, you can see that the nuclear envelope breaks, the centrosomes, they replicate and start moving to the opposite poles. In metaphase 2, the chromosomes line up along the equator, and now their homologous pairs are not there in the cell at all. The chromosomes align along the equator and the spindle hold the chromosomes at the region of centromere through the kinetochore. And there are no homologous pairs right now because the homologous pairs have been separated. So each chromosome is a different chromosome. 23 will align here, 23 will align here for a human cell. This is followed by anaphase 2, where the spindle retracts again. As the spindle retracts, the sister chromatids are pulled apart. And when the sister chromatids are pulled apart, again the centromere will be going towards the centrosome first, and these limbs of this chromosome will be dangling along. So the sister chromatids have been separated. Here the centromere splits. Telophase 2. So in telophase 2, you can see 4 haploid cells forming, each having the chromatids separated from the other. Nuclear envelope reforms around each haploid nucleus. The nucleus is still haploid. At the end of meiosis II, 4 haploid cells form. All the 4 daughter cells are haploid, each having 23 DNA molecules on 23 chromatids, appearing as 23 chromosomes. So the DNA content reduces in such a way that the cells here, they end up receiving 23 DNA molecules, something that we discussed initially. This is how a gamete is formed. If you are given this photograph, arrange the following stages of meiosis in sequence. So probably you can just work this out. You can see that this is prophase 1. You can see the chromosomes here, and you can very well see their appearance. This is the bivalent pairing up in E. So this is middle prophase 1. This is crossing over occurred here. So this is late prophase 1. Here you can see the homologous chromosomes. They have paired up along the center. So this is metaphase 1. Here in F, the homologous pairs are being pulled apart. This must be anaphase 1. In D, 2 separate cells with no homologous pairs can be seen. So this is cytokinesis, which has followed telophase 1. In G, single haploid nucleus can be seen and single haploid cells can be seen. So this must be prophase 2. In I, the chromosomes line up in the center, no homologous pairs. So this is metaphase 2. In C, you can see sister chromatids being pulled apart from one another. This should be anaphase 2. And 2 separate haploid cells with single chromosomes and single chromatid can be seen in each nucleus. This is cytokinesis of meiosis II. So this sequence can be seen. Now, what is independent assortment of chromosomes? How does this concept align with meiosis, and how the alleles and the chromosomes, they separate out by independent assortment? Genes for the 2 traits, for example, stem color, capital A small a, and leaf shape, capital D small d, are located on 2 different chromosomes, on 2 separate chromosomes. They would follow independent assortment, and the genes are not linked with one another. When meiosis occurs, both genes and their respective alleles, they would assort independently. 4 types of gametes would form in this case by the end of meiosis II. So this is how the cells look like initially. You can see that these cells can be seen to be in the first one is in prophase 1. You can see the chromosomes there, and this is late prophase 1, in which the chromosomes have come in pairs. So one green for the other purple for the other, this orange for the other orange. On this chromosome, this is capital A allele present. On its homologous partner, the allele small a is present. On this other homologous pair, this is capital D, this is small d. So capital A small a chromosome bivalent pair up, capital D small d bivalent can be seen pairing up as well. I'm not showing any crossing over here. Metaphase 1, the chromosomes line up in the center and they line up as pairs. So they can line up in such a way that small a is here and the capital A is here, small d here and capital D here. Or they can line up in such a way that capital A and small a are here, very similar to this, but the capital D is at the top and small d is here. It's rotated. So they can arrange in these two ways. There is no third way for these two alleles at least, or these two traits. Now, anaphase 1 occurs and these alleles get pulled apart, not pushed apart. So what will happen in anaphase 1? This is what was already there in metaphase 1. So this is the diagram continued from the previous slide. This is the stage going in anaphase 1. So what will happen in anaphase 1? These will be pulled apart. These A's and D's will go down. This A and D will go up. This A and D will come down, and this A and D will go up. So by the end of Meiosis II, 4 type of different gametes will form, and these 4 gametes will have small a and small d together, or capital A and small d together, or a small a and a small d together, and a capital A and a capital D together. It all depends of where the split occurred. So when the split will occur, this chromatids, they come together or here or in this way or in this way. So the 4 different gametes or 4 different type of gametes can form. So the carry home message from this is that if 2 organisms who are both purebred for the 2 traits, for example, capital A, capital D, and small a, small d were crossed, all the F1 offsprings will be 100% hybrid, which means capital A, capital D, small d. So that can be seen. When you cross the F1 offsprings together, you get 2 hybrids being crossed together, the 2 offsprings of the F1 generation. The ratio of the offsprings now will be 9:3:3:1 ratio of offspring, with 2 recombinants, which means 2 new varieties coming in the phenotype, which will not be alike this parent or this. So they will be new varieties, recombinants altogether. These hybrids can be used for another cross, the crossing of F1 hybrid offspring, which is capital A, capital D, small d, with the purebred recessive small a, small d, small d. Now 1:1:1:1 ratio can be expected, because here 2 recombinants can be seen, 2 new varieties can be seen, and 2 parental forms. These ratios, if they are seen, they show that the genes capital A small a and capital D small d are not linked and they sort independently during meiosis. I hope that video was helpful. Do let me know, was it helpful or not? Thank you so much. That's it from my side.